Okay. All right. Well, thank you. Thanks for coming out on a Saturday. And I hope the new hotel is better than the old one. Yeah. Okay. So today's talk is going to be a little bit different. It's not going to have nearly as much math in it, or even sort of specific data that I'm going to talk about. In fact, it's even going to wander a little bit from theory over into what you might call philosophy. And if you remember, at the panel discussion that we had at the NCBS, there was this discussion of, whether theory gets any respect. Well, if you think theory doesn't get any respect, you should try philosophy. Most people think philosophy is just, sitting around in armchairs and musing about the larger questions in life. But in fact, philosophy plays a big role in what we do, because philosophy is really just a statement of what are the organizing principles that we use so that we can take a principled approach to how we approach science, right? So, and that's actually important, particularly when, like now, things are changing very fast in terms of what we work with, right? To sit back for a moment and think about, what are the principles that guide how we should interact with our material and our data and so on. I think that's something that's worth doing. And I particularly wanted to talk about this topic because, even though from a sort of theoretical or physics point of view, this may not be the most interesting topic in the world, from a practical point of view, I think this is the driving topic in systems biology right now, is how to handle big data. And so in terms of, where people are going to be scrambling to work and where the money is going to come from and what the public is going to be interested in, I think a lot of that is going to fall into this arena. So I think it's a good idea that we spend some time thinking about it. Okay, so today's talk is very much focused on thinking about things. Okay, so let me start with this nice picture that is representative of a parable that, as far as anyone knows, originated here in India, which is the parable of the blind man and the elephant. So how many people are not familiar with this parable? A few, okay, all right. So this, there's a famous poem by an English poet on this, but this is actually a drawing from a Japanese artist of blind monks examining an elephant. And the point is, if you're blind and all you have is what you touch and feel as your way of knowing something, then everyone is going to get a completely different impression of what an elephant is. So to this man, an elephant is essentially a sword. And to this man, the elephant is a wall. And to this man, the elephant is a brush. And, because each one only feels part of the elephant. And they're all wrong and they're all right at the same time, right? And anyway, so it's a parable about how we sort of construct our world out of the little bits that we interact with, and that there's, certain dangers inherent in doing that. Certainly we won't agree very much if that's all we can do. So another way to put this, if we try to relate this to science, where I think the metaphor of the blind man and the elephant is very appropriate, is to say that science progresses by testing hypotheses. But hypotheses don't come, ex vacuo. Hypotheses come from somewhere, and they only come from what we already know. And there's another parable that's it's told a lot in the US. I don't know how common it is here. But I know when I was a graduate student, people used to tell this tale all the time about the drunk in the lamppost. How many people have heard this one, about the drunk in the lamppost? A couple, okay. So the idea here is that, there's a policeman who noticed a man outside a pub, right? And he's kind of on his hands and knees feeling around, and he's obviously drunk. And the policeman asks him, says, well, what's up, sir? He says, I'm looking for my keys. I can't find my keys. And so the policeman stoops down and looks around too. And after 5 or 10 minutes of not finding the guy's keys, the policeman says, "Are you sure you lost them over here?" And the drunk says, "Well, no, actually, I have no idea where I lost them." He says, "Well, why are we looking here?" He says, "Well, that's because that's where the light is." You know, and it's funny because as a graduate student, I was always told that this is actually a parable about how you should approach science, right? That you should look where there's light, right? There's no point in working on something where nothing else has been done because practically speaking, you know, it'll be too hard. And so you should always look where there's light and build hypotheses just a little bit outside of where there's already a lot of knowledge, right? So that was kind of given as practical advice. I don't know if people still give this advice to graduate students, but it used to be very common to do so. And so we did that, right? That's what we've been doing in biology for a very long time. And, you know, the consequence of that is we've ended up building up a picture of biology that's come from looking where there's light, right? Taking the information we have, using it to create new hypotheses, adding on to it. And, you know, as a result, we build up pathways, right? And I remember back in the mid-1980s when I was, you know, a postdoc, you know, we had these lovely pathways that had been built up by adding one hypothesis onto another, like the EGF receptor pathway, which starts here clearly, ends up there at the transcription factors, and has these nice phosphorylation and binding steps in between. And we kept doing this, and of course now, by say 2008, the pathways are all starting to look like this, right? And in fact, in 2013, it's even more complicated than this, right? So by this point, people begin to wonder maybe there's kind of something wrong with what we're doing, because I think it's fair to say that in 1985, we understood the EGF pathway, and in 2013, we don't understand the pathway, so it seems like we've gone from understanding to not understanding, which doesn't seem like the right direction to be going in. But this is really, you know, what's happened all over biology, right? We've suddenly realized we are the blind men, and biology is the elephant, and whatever we do, we're feeling just a little piece of it, and we're trying to construct the big picture out of it, and we're struggling to do that. And so now everything is networks, right? You know, you have networks of gene expression, and networks of protein-protein interaction, and networks of genetic effects, and everything, people are sort of scrambling to say, well, it's all huge interconnected networks. And so essentially we've gone from the previous era, you know, where the icon was the DNA, the double helix, right? This beautiful orderly structure was kind of our metaphor for biology, and now the icon is the hairball, right? And that really is, you know, you say, well, what's biology like? Okay. And so the problem is a hairball is not an elephant, right? Not lately, at least. Right? And so the question we face in biology is how do you get from the hairball to the elephant? And that's a new question. Right? I mean, it's not something that couldn't be posed before. But what's new is that everyone is forced to confront it now, right? Because we're filling up with hairballs. And we know that what we really want is elephants. Okay. So a lot of people are motivated to try to do something about this. Right now in the world, a large number of people feel that the appropriate way to do something about this has something to do with big data. And you know, the notion of big data didn't start with biology. It started with physics. It started with Google, and you know, lots of social networking stuff. I mean, there's a lot of enthusiasm for the power you have to do useful things, or at least make a lot of money, if you have at your disposal really, really big terabyte, petabyte, you know, all this kinds of big data. And so you see in systems biology, you know, the human genome and other genomes and metagenomes, transcriptomes and code projects, the Cancer Genome Atlas, personal genomes, genome-wide associations, phenomes, electronic medical records, metabolomes, databases of models, databases of proteomes. And then the latest thing in the US is this brain mapping project, right, where they're going to spend a lot of money to make a map of every synapse, right? And, you know, and a functional map of every synapse, right? Yes, I know. Exactly. Yeah. Well, you can fit a lot of data on a picture, right? So you don't need 10 to the 12th pictures. But the point is exactly what you said. It's like, has anybody thought about the, the ramifications of what they've asked for? And yet, that's what people are asking for. If you know, in the States or in Europe, the funding agencies have released, you know, their calls for what they want people to do, and big data is right there in it now. It's like, you gotta get that stuff. So what's the promise of the big data, right? So the promise is if you take the hairball and you can find the right correlations in it, and you can apply the right algorithms, and you can do machine learning in the right sorts of ways, you will be able to get the elephant, right? You can reconstruct biology out of this big data. And the reason why that's exciting to people is it seems like it solves this problem, the prior knowledge problem, right? That in the old days, we could only build a hypothesis from what we already knew and could only thereby move slowly across the elephant. This is a way of saying you can do the whole elephant at once. You don't have to look where there's light, right? You can illuminate the whole parking lot, right? And thereby, you know, cover everything, be comprehensive. And that will avoid that problem of the blind man and the elephant, and you'll be able to see the big picture. That's the promise of big data, that there's an algorithmic way from big data to the big picture. So the question that you know, I'm focusing on is that really true? Is that really feasible that you can get from here to there? And so in thinking about that, one of the first problems that comes to mind is that, there's the idea, right? First problem that comes to mind is that, you know, if you let the data do the talking, how do you know which data to listen to, right? I mean, can you really listen to them all at once, or is it like, you know, a cacophony, right? Is there some need to actually assess which data are important? And so I would argue that there is, right? I'd argue that it's absolutely essential when you're dealing with a hairball like this that right at the outset you'll be able to identify some subset of the hairball that's important, okay? Because otherwise, you know, you're going to lack certain key things, right? So why do you have to sort of pre-identify importance when you approach a big data set? So I think there are two reasons for that, two separate reasons. One has to do with the meaning of prediction versus the meaning of understanding, right? There are many people who sort of reflexively will say, well, if you have predictive power, you understand the system, okay? And that's confusing a definition with a correlation, okay? When you have predictive power, that might be a sign that you have understanding of the system, but it's not understanding per se. And one way to think about it is as a thought experiment. This is what philosophers like to do, right? I mean, imagine you had a computer database, kind of like a super Wikipedia, and it had the answer to every question you could possibly ask. It was in there, kind of like that machine in the Hitchhiker's Guide to the Galaxy, right? The answer to life, universe, and everything. Anyone remember what the answer was? 42. There, okay, good. So suppose you had such a machine, and it had the answer to every possible question. Therefore, by definition, you now personally have perfect predictive power, right? So the question is, do you understand anything? Not really, right? I mean, you might, but there's no reason why you have to understand anything because you have the perfect predictive machine, right? So that's just a way of saying that having predictive power isn't the same thing as understanding something. So if that's the case, well, what is understanding something? What does it mean when we say, I understand this problem? I've explained the problem, right? So there's a lot of ways people try to define this. This is the one that I find the most compelling, that sort of fits the best with examples in science, is that to understand is to possess a process. You can call it an algorithm or a method or a heuristic that systematically produces, you can call that predicting or retrodicting or just corresponding to, a bunch of facts starting from something much simpler than the facts themselves. So that's why just having a computer that has all the answers stored in it is not understanding. But if you have something that doesn't have the answers, it has something smaller, some algorithm that will produce them, right? And if you possess that, then you can say, well, yeah, I have some understanding. And a classic example, right, is the laws of physics, right? Or some of those examples that Rob talked about of data collapse, right? When you can make the data collapse on one curve, you have an understanding, because you can basically say, this isn't just a pile of data. This is this one equation. All you have to do is scale it a little differently, and you get the data. So to me, that's the essence of understanding, is to be able to make a much simpler version or model and show that it works. Okay. So in a sense, understanding a problem is inseparable from the notion of stripping it down to what's important and what isn't, right? And so therefore, I would argue that if you're going to understand biology from hairballs, at some point, you've got to get to be able to answer what's important with respect to whatever part of biology you're interested in. If you're interested in fitness, or if you're interested in metabolism, or, right, for whatever you're interested in, you need to be able to say, this is the important part of the hairball. Okay, that's one reason why importance is important. The second reason is a much more practical one, but a very thorny one. And that has to do with the fact that biological data are noisy, and only some of that noise is measurement error that can be improved. A lot of the noise is biological noise, right? And so consequently, we can only really assess things and test predictions in a statistical sort of way, right? We can only say with X probability, this thing actually turns that thing on, right? And the problem is when you start with a pile of data and you're trying to make inferences out of, 10 to the 6th pieces of information, and you can only say things in a statistical way about each of your inferences, you have what's called the multiple hypothesis testing problem, right? Which is that, if you asked each inference to merely pass the test of, has a 5% chance of being wrong, then among the whole pile, the number that are going to be wrong is huge, right? So you have to require a much more stringent statistical criterion. The classic example of where this arises is in genome-wide association studies. So did Boris talk about genome-wide associations at all? No. Okay. So, but this is just a very simple practice of, you take a bunch of people, or yeast, or whatever you will, and separate them into those that have some phenotype that you're interested in, and those that don't. Maybe these have diabetes or cancer or whatever, and these don't. And you collect their DNA, and you essentially genotype the DNA. Soon it will be by fully sequencing, but still to this day, we mainly do it by markers that are placed frequently along the DNA. So you can figure out who's got what alleles, and you essentially do a correlation, right? Just a sort of first-order correlation between which alleles are more or less likely to be correlated with having the phenotype or not having the phenotype. Okay, and then you do that for all of your possible markers, and you look for which ones score as correlated with a p-value, and you can see what some of these p-values are. Log 10 of the p-value is 10, so 10 to the minus 10th, right? And the reason you need that is because you're comparing so many samples, right, that only something that has a p-value that high actually has a reasonable chance of not being an association by mere chance. And so typically, in order to have enough coverage of the genome, you have to assess about 1 million of these variants across the genome. And, right, so you have a multiple sampling problem of 1 million, which is why you have those very high p-values. But what if you were interested in, do genes in pairs cause some phenotype, or in triples? Well, you have 5 times 10 to the 11th pairs and about 10 to the 17th triples. And it's very easy to show that even at this point, there are not enough human beings on the planet to be genotyped to give you the statistical power to find any of those associations, right? So lots of things which in principle you could infer just by correlations. In practice, you can't because of all the biological noise, right? But on the other hand, if you knew a priori what was important, right? If you knew right away what was important, then you wouldn't have to do so many comparisons, right? The reason there's so many comparisons is this is what's called a hypothesis blind experiment. We're assuming that any possible gene or allele could be involved in a process. Okay. So how do we find out what's important, right? Now that I've argued that even before you dig into a pile of big data, you have to have some sense of what's important, or at least have some way of finding out what's important. Well, so one way we can ask that question is look at the historical record. In the history of biology, has anyone ever faced this problem before? Where you have a system, you don't know anything, and you want some algorithmic way to discover what's important. Okay. Well, we do. That's the field of experimental genetics, right? That's the beauty, or as some people say, awesome power of experimental genetics, is that you can approach your organism and do nothing but define the phenotype that you're interested in. No wings, grows fast, resists antibiotic, whatever you like, and then you can do what's called a genetic screen. You can make mutations all over the genome and then recover whichever animals have that phenotype. And then figure out which gene has the mutation in it, and bingo, you've associated the gene with the phenotype. So that's a way of saying that gene's important. But let's be really careful about our language here, because actually what that is, it's a way of saying it's an influential gene, right? You're saying this gene, when mutated, produces that phenotype. So this gene has an influence on that type of phenotype, okay? And what you're doing is you're saying, operationally, I will define it as important if it has an influence on the phenotype. So it gives you an operational way of saying, here's how I'm going to figure out what's important, and then I'll work with those, right? And back in the 70s and 80s and early 90s, people were very enthusiastic that through saturation mutagenesis, saturation screens, where you basically keep screening until you've covered every possible mutation that, not every possible, but enough that you've hit most of the genes of the genome, that essentially you'd be able to find all the things that were important in all the phenotypes you might be interested in. And several Nobel Prizes came out of really influential saturation screens in the fruit fly, for example, that picked out all kinds of really cool genes involved in pattern formation. Or in C. elegans, that picked out lots of genes involved in cell death and also in development as well. And there was this sense back then that it was just a matter of time before the experimental geneticists solved everything. In fact, if you ever heard this guy, Bob Horvitz, give a talk, he used to go around back in the late 80s and early 90s basically telling everyone to quit working on any organism other than this one, because they knew all the cells, all the genes, they'd saturated the genome with mutations everywhere, and it was merely a matter of time before all of the major questions in biology were going to give way. Well, obviously that didn't happen, right? So what happened after this to sort of change people's minds that you could get to understanding of everything just by doing saturation screens? Well, one thing that happened is geneticists began to realize you can go in the other direction. That is, once we began to have genome maps and later genome sequences, it became clear you can not just randomly make mutations and look for things that have certain phenotypes, but you can just go gene by gene and knock the genes out and see what phenotypes you get, right? So that's called reverse genetics or targeted mutagenesis. And right away, people got a little bit scared with what they found, right? Like in the yeast genome, less than 20% of the entire genome is actually essential. Everyone was surprised by that. It's even worse in the mouse. Most vertebrate knockouts have, quote unquote, no obvious phenotype, right? And when knockouts do have a phenotype, it isn't necessarily the case that you should even consider the genes to be important, right? They can have a phenotype because something happened unrelated to the process you're interested in. You had a question? Yeah, so obviously that's the key thing here, right? Essential is related to, it's an operational definition. It's saying if I grow the yeast in this condition, does it live or not, right? You could define the operation differently. You could say, could the yeast outcompete another yeast? That's very different. Could it grow on minimal medium? That's very different. And the same thing with the mice. Some of these knockout phenotypes that have no obvious phenotype will have a phenotype if you look at it in a different way. So I'm not saying these genes are unimportant, right? I'm just saying that if you use this very simple operational definition of can you connect it to the phenotype, it's not coming out as often that way as you'd expect it would. Yes. Yeah. Yeah. Yeah, sure. Essential under a certain condition. But these are yeast. I mean, they're not really developing, right? They're just growing. Oh, you mean in this case, in the mouse. Right. So it's not the fact that many of them are lethal. So let me make sure that you're not implying that we're missing the lethals here. No, no, no. We see the lethals. Those are easy to find, right? Because, right. It's just not as many as you'd expect. I remember when they knocked out Sark, which was this gene everybody worked on because of how important it was. And the Sark mutant mouse looked perfectly normal. And people were freaking out, you know, ready to jump out of the windows because it just didn't make sense, right? And actually, the Sark mouse does have a phenotype, but it's not at all related to what you thought it would have been related to. So when reverse mutagenesis came along, right, people began to wonder whether the understanding they'd built up from saturation screening, you know, was the right one, because it wasn't corresponding here. It got even worse when people started doing serious population genetics in the molecular era. So these are two flies having a fight. And believe it or not, fruit flies have fights. They can be aggressive, and you can select for aggression. But in population genetics, rather than mutate the genome, you just start with wild populations that already have different standing variation, and you cross them together, and you select for a phenotype. And it's well known to fly breeders. Actually, you can select for almost any phenotype in a fairly short time in flies. So this is work from Ralph Greenspan, where he selected for aggression 21 generations with lots of intercrossing and stuff to make sure that things weren't linked as badly as they would. And then he looked genome-wide at gene expression in the head. And that was because at this point in time, it was too hard to sequence everything, but gene expression was a proxy for the fact that there were allelic differences here. And he found 45 genes that had significant alteration in expression, not very large, but significant differences in the head, which is mostly brain in his case, in these flies. But the surprise was 8 genes had been found by saturation mutagenesis to cause this phenotype. None of them were on his list. Right? There was 0 overlap. Now, if you don't like aggression, maybe you like a milder phenotype like wing shape. Okay, so wing shape is another selectable trait in flies. Okay, and you do the same kind of experiment, right? You select over time for flies that have one extreme of shape or the other extreme of shape, and then you compare them and you figure out which genes have expression suggesting they have something to do with the shape change. You find 27 genes. There are 214 genes known to control wing development from saturation mutagenesis. Not one of them is on that list. Okay, so all of these genes are somehow connected to this phenotype, but they're not the ones you come up with saturation mutagenesis. And actually, if you do the experiment again, you get even more genes that are not on this list and not necessarily strongly overlapping with that list.
Could I interrupt? And, looking at that as an ex-geneticist, I would say there's something wrong with the significance of the things they're looking at. Like they've done microarrays and find a twofold effect. And
Right.
just shouldn't have believed.
Well, so in that case, it's still 0, right? You're not finding any of the things you're supposed to find. Even if you don't like these, you're still not finding.
It's different than the strong
Yeah.
claim, though, that
Right.
every time you do it in a different lab, you get a different set.
You don't get an entirely different set, but yeah, okay.
Looks like these are two different questions. So the genes that control development maybe in the adult fly switched
No, no. These genes are known, many of them are known to affect this phenotype in the adult. Yeah. The expression wasn't measured in the adult. The expression was measured during development when these patterns are, yeah, that's actually a good point. Yeah.
It appears to me that the rewiring, the reprogramming hasn't been considered explicitly. So this could be an effect of the knockout of that, but we are not even looking at that.
Could well be, yes. Right, it could be that, you know, but I mean, it's part of the same picture. It's just when you do the knockout, it's like the blind man and the elephant.
Yeah.
There's one way of constructing this phenotype. And this is just saying there are other ways to construct the phenotype. Let me just show you one more example, which is a little more modern. And at this point, there's actually sequence data so that you can actually look at the mutations and so on. And this was work done by Tony Long and Michael Rose, in which flies were selected for accelerated development and early fertility for 600 generations with multiple lines and lots of mixing. Anyway, they expected to find single genes selectively swept to high frequency in these populations. They never saw that. What they saw was that the allele frequencies changed at 506 genes, in most cases repeatable from line to line, so most of that's not noise. Some of it may be linkage, and therefore, you know, they're not necessarily involved. But the interesting thing is, you know, only a quarter of these have any developmental phenotype as learned from knockouts and saturation mutagenesis. And not only do only a quarter of them have any developmental phenotype, as far as I know, none of these genes have ever been associated with this phenotype. So again, it just suggests you get different kinds of genes right, when you approach the problem one way as when you approach the problem a different way. Another way of saying this is influence is an operational definition, right? Your definition is, if I change that, does that happen, right? And then you can say, yes, it does, so this has an influence on that. But it's not the same as importance, because depending upon how you do the operation, it doesn't seem like you get the same ones. And so which one really is important? If you think about the databases we turn to, the KEGG and the Ingenuity and all the, these are mainly assembled by certain classes of experiments used to implicate these genes as being the important players. Can we be confident that that's really capturing what's important? So, in this regard, I was really amused when Rob gave this analogy in his discussion at the panel of the Airbus seatbelts, right? So he said basically if he removed or broke the seatbelts on an Airbus 380, it would never take off, right? And so by any measure of influence, these are important in flight of an airplane. And so therefore you should go and measure the spring constant, right? Which is, but that was his point, right? He was really making the same point, that just because something is influential in an operational test, it could have nothing whatsoever to do with what you're testing, right? Does the airplane fly or not? Okay, all right. So now at this point you might think, oh, he's being a pain. He's just like coming up with these rare examples of, straw men and, So is this really a problem or not, right? I mean, I've said that being influential is not the same as being important. But isn't it still true that most of the time you're going to get the right things, whichever operational test you do? So I want to convince you that the answer to that's no, okay? And I want to convince you first with a couple of examples. And the first example has nothing to do with biology, but it makes the point well, and that's the noise-canceling headphones. Everyone's familiar with noise-canceling headphones, right? I mean, I couldn't survive an airplane flight without noise-canceling headphones. So now, when you put on the noise-canceling headphones, that enables you to have some music source, and the performance is that it produces music that you hear, right? That's what you want this thing to do, is enable you to listen to your iPhone or your iPod or whatever. Okay. So let's approach this like a biologist, and let us find out what's important to this circuit. So we'll take away the music source, no performance. Obviously, that's important. Take away the battery, no performance. Obviously, that's important. Screw up the fit on the ear, performance is degraded. So obviously, that's important. Take away the electronic circuitry, no performance. That's important. Take away the noise. What happens? Nothing. Performance is just fine, right? So what will you conclude is the least important part of this network? The noise. But they're noise-canceling headphones, right? From the perspective of what they are and their function, right? The most important thing is noise, right? If it weren't for noise, you wouldn't need these things. And yet, by any operational test based on their performance, you will not see the fact that noise matters. Why? Because they're noise-canceling headphones. Okay, you can see the same thing in biology. You know, we have this very elaborate system that maintains the salt concentration in our blood very steady, you know, like 135 millimolar, whatever. Anyway. And it's very, 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 very good system to keep the salt there. And so consequently, if you go out and eat a salty meal, it doesn't matter. The salt in your blood stays pretty much the same, right? Very small changes. If you knock out the hypothalamus, your salt goes crazy. If you knock out the pituitary, the antidiuretic hormone, if you mess up the kidneys, right, change the water, all these things will affect your salt. But if you take in too much salt, nothing happens, right? It's the same thing. It's a salt regulatory system by any sensible means. Salt is the most important thing to it, and it's the one thing you can't identify unless you already know to look for it. Now, if you know to look for it, you can say, oh, well, great. If I take this out and then challenge with salt, now I'm going to see a big difference, right? But that's cheating, right? That's not hypothesis blind. That's already knowing what it is you're going to be looking for. So the feature that these systems have in common is they're feedback controllers. Remember, a feedback control is where you have machinery that has performance. It can be perturbed either up or downstream of the machinery. You know what the optimal is, and so you can generate some kind of control signal and nullify those perturbations. Right? And it's because the system is designed to nullify the perturbations that your ability to show that those perturbations are super important goes away. Right? Okay. Interestingly, natural selection itself can be thought of as a type of feedback control, right? On a very long time scale. Right? You have genotype, that's the machine. You have performance, environment can affect it. Mutation can affect the genotype. And the comparison of performance with optimal performance, I mean, there isn't really an optimal, but the comparison is what we call fitness, right? And then fitness selects different mutations. And so the same loop can be drawn, right, for evolution. And so that suggests that, that the feedback loop of natural selection is also going to make certain very important things very difficult to find, right? Because it's essentially a control system. And we've become very interested in what we think is one case of this, which has to do with the tendency of natural selection to select for genetic robustness. So I have to unpack this a little bit. Remember, robustness, right, is the ability to achieve performance that's relatively insensitive to various kinds of perturbations. So genetic robustness is performance that's relatively insensitive to mutations. Okay? We, in fact, have a great deal of genetic robustness. We often don't think about it, but we're diploids. We have 2 copies of everything. And for about 95% of our genes, we can take 1 copy away. Nothing happens. Right? In fact, most of us have a missing good copy for, it's estimated, at least 7 of our genes. And 7 really essential genes, right, that we're missing at least 1 good copy. Why is that? I mean, isn't that really bizarre? If you're an engineer, imagine someone said, go build a device in which every component of the device, you can reduce its performance by half, but the performance of the whole device shouldn't change at all, right? I mean, very little that we engineer has that character, but our bodies do. Yeah, so you're saying, are these copies redundant? Well, I'm not sure I'm comfortable with the term redundant, because there are so many assumptions that go into even using the term redundant. But what you're saying is, yeah, sure, it's possible to build systems in which the quantitative level of something isn't really that important. It's just the presence is all that matters. But there are some systems that are intrinsically quantitative, like these development systems we talked about where you're controlling pattern, right? It's the morphogen's supposed to be at that level at that point, right? So that's unlikely to be just that the system is binary. You just need presence or absence. So, okay. Anyway, so it's a fact that you know, you can tolerate mutations in a lot of genes. Question is, why does that arise? And there's been a lot of theorizing about the evolution of evolvability, and you know, but really there's a much better answer for that, is that it arises because it has to arise because external perturbations, environment, changes the levels of expression of things. That's what environment does. It changes the level of expression of genes or the level of activity of enzymes or so on. And there's obvious reasons why you'd want to be robust to that. Right? So evolution is going to select for robustness to environmental perturbation. And the problem is once you do that, you automatically gain robustness to genetic perturbation. And that's actually all this graph shows. This is Kuni Kaneko in Tokyo, who just built these big models that are generic gene regulatory network models. They basically have sort of switch-like behavior and then a noise term that represents environmental perturbations. And he'd build these models populated with whatever parameters he'd like, you know, with many different equations, and then he'd throw in noise of various sorts, and then he'd evolve them so that their performance was better in the face of noise. And he found that as he decreased the variation due to external perturbations, he automatically ended up decreasing the variance, in this case, due to parameter perturbations, which would correspond to the sort of values of the genes. So this is just sort of a mathematical way of showing that if you optimize for resistance to noise, you end up optimizing for robustness to your parameters. Okay, so it's not an accident. And sometimes in evolutionary biology, that's given the name survival of the flattest. Okay, and the reason for that is because if we represent the phenotype as some value and the gene activity on some axis, right, then being robust to changes in gene activity correspond to having a flat position on that landscape. And you know, traditional evolutionary biology math tells you that, you know, this organism is equally fit to that organism, but the reality is this organism is more fit in a fluctuating environment, right? And so that's what we mean by survival of the flattest. Actually, recently I saw some really beautiful evidence for robustness to mutation in a real developmental system. This came from Marianne Felix from Paris, who works on C. elegans and on the development of the C. elegans vulva, which comes from these cells here. And the cells adopt different fates in a very characteristic way. And there are a bunch of phenotypes you can get if you make mutants. And I'll spare you the details, but the phenotypes are, oh, there's some words here that didn't come out. It doesn't matter. The phenotypes are, she gives them categories A, B, C, and D. These phenotypes almost never arise in wild populations, or if you look in different strains or compare even across different species, these are very, very rare phenotypes, even though they're not lethal. This other category, which I think involves the fate of that cell, happens, but pretty rarely, both within and between species. And then this fourth phenotype, which involves the fate of that cell, is a very frequent thing to vary from one species to another or from one isolate to another in the same species. So it turns out that, that Michael Lynch has developed lines of C. elegans in which they basically let them go through a generation, pick one animal, restart, go through another generation, pick one animal. So you go through many generations, at each generation just allowing one animal to survive. And over hundreds of generations, you're waiting for mutations to accumulate. And so those are called mutation accumulation lines, and there's several hundreds of them. And because there's no selection going on here other than selection for viability, right, the mutations levels are just reflecting the rate at which mutations are happening, right, as opposed to you know, what phenotypic effect they're having. And when you do that and you look at these phenotypes, you find that mutations much more frequently cause this phenotype, at a low level cause this phenotype, and never cause that phenotype. Just like what you see in standing variation. So basically it's saying, if this argues that this is under selection, right? And things that are under selection are also much harder to reach by mutation. Things that aren't under selection are very easy to reach by mutation. Which is another way to say that selection selects for genetic robustness. Okay. Now, of course, phenotypic landscape, yes?
Isn't that the more the system becomes robust, it loses its capability to evolve?
Yeah, we're getting into philosophy here, right? Yeah. So there is definitely trade-offs between robustness and evolvability, right? And so, you know, if you had something that was so robust to every mutation that no mutation could change the phenotype, well, then it couldn't evolve, right? But those are, you know, extreme conditions, right? The reality is nothing is perfectly robust, and mutations can occur that take you to non-robust regions. And as we'll see, there are other issues like combinations of mutations, which we'll talk about momentarily.
Can we use this argument reverse, like if some protein is very conserved, can we say that the problem it is solving is very robust? Like
well, not the problem is robust, but yeah, if you see a protein that's sequence is really, really highly conserved, you would argue it's likely to be under selection, right? That there's, right, that it matters that it has that sequence. And so you would predict here, right, that also, you know, it would be more mutationally robust as well because it's under selection. Don't know. I mean, the example I showed is actually one of the prettiest I've seen in a while, but it's again, you can just make the argument based on the heterozygous effect that, you know, we can be heterozygous for so many genes. Okay. But the point I want to make here is that, of course, I drew a phenotypic landscape one-dimensionally, right? There was just one gene and there was a landscape. But, you know, the reality is our genome, our phenotypic landscape is like an n-dimensional version of this, right? There's just peaks and valleys in multiple ways. And, you know, so all of these would represent phenotypes that are the same, right? But some will be more robust than others. And so selection will tend to move you around on this, you know, plane within your phenotypic landscape. When you think about that, you realize that robust doesn't necessarily mean survival of the flattest in every direction. It really means survival of the flattest along the axes. And the reason for that is because of the sort of mechanics of natural selection. If a single gene, if mutations in a single gene have a phenotypic effect, it's relatively easy to select for or against that. But if it takes a pair to have the effect, it's much harder. That is, it takes much more evolutionary time to select for or against that, because you have to wait for that pair to arise, and then there's all the times when those alleles occur not in that pair, right? And there's no selective pressure going on there. And of course, with the triple, it's even worse, right? And so on. The more combinatorial, right, the less this selection for robustness, whether it's because of environment or whatever, just the more combinatorial it is, the less effectively one can select for robustness. And so consequently, this landscape is perfectly reasonable, right? You could select for this one easily, but you wouldn't select for that one, even though overall this is much flatter, but it's less flat along the cardinal axes. Okay, so geneticists or evolutionary biologists have realized this before. This is not new. And it's been pointed out in a different way. which is, so epistasis is a term used essentially to mean nonlinearity of interactions, of effects between different genes. If it's not additive, you call it epistatic. It can be synergistic or antagonistic, right? And it's been pointed out that as you select for robustness, you build up epistasis. That is to say, there are more cases in which interactions have a bigger effect than if you didn't select for robustness. And it's really a relatively kind of simple geometric argument, right? Is that when you select for robustness, you're failing to get rid of cases like this. So this case has strong epistasis because the phenotype change is huge between the axes, but hardly at all, right, on the axes. So this is a very non-additive effect. This is much less epistatic, right? So the idea is by selectively enriching for things that are flat on the axis, you end up disproportionately enriching for things that are non-flat between the axes, okay? And so that's this argument about selection for robustness increases synergistic, in particular, synergistic epistasis. And usually that's measured by some measure of average epistasis in the population, which, you know, is some number that it doesn't undergo a huge change. But we sort of felt like this wasn't, you know, from a practical standpoint, this was maybe not the right way to look at it. Because what we're interested in is not what average epistasis occurs in a genome, but rather when you have a phenotype, some extreme phenotype like, you know, diabetes or a birth defect or whatever, you want to know, was epistasis responsible for it? So you want to know about the outliers, not about the mean, right? And so we thought we would just take a very dumb brute force approach to that question and just take a bunch of models out of the literature. There's this model database where you can just download other people's ODE models and Boolean models and things and then implement them and run them. So we just took a bunch of models, circadian oscillator model. This is more of a randomly connected gene regulatory model, cell cycle model. These are kind of classic systems biology models. And we said, all right, well, let's, you know, randomly initialize those models with some parameters. In the case, if the model came with what it considered were the best parameters, we use those. Right? In other cases, we just pick them at random. And we defined the task that the system, the input-output relationship, often it was a multi-input, multi-output relationship. But we defined it. We said that's what we're going to say the system does. Measure the robustness to small perturbations around the parameters. And then just mutagenize these models for a while until they got more robust to their own parameters. The parameters being analogous to the different genes underlying, say, a genetic network. So here, for example, is a circadian oscillator. I know one of the groups is doing circadian oscillation. This is one of the simplest circadian oscillator models, just a few differential equations. It has the character that it oscillates when entrained by light pulses, right, which come in over here. And also, if you shut down the lights, it continues to oscillate at the same frequency, right, for a very long time. Okay, and so we, you know, demanded that it do all these things and that, you know, the amplitudes don't change too much and so on. Before we did anything to the parameters, this system, which is a very nonlinear system, shows some very high sensitivities to parameters. So remember, these are these sensitivity coefficients. So a value of 2 means the output goes with the square of the parameter. 3 means it goes with the cube of the parameter, right? So there's some pretty high sensitivities there. Interestingly, if you look in the initial model before any evolution, you say, okay, well, what happens if we take parameters and perturb them pairwise? Can we see the nonlinearity? In fact, you generally don't. You generally see that the observed effects fall close to the line of the predicted effects. And where they fall off of it, they tend to fall on this side, which is the antagonistic epistasis side, mainly meaning that the effect of doing 2 things is less than you'd predict from just adding them up. And mostly that's because of saturation, that sometimes the one perturbation already takes you, you're essentially broken. You can't break any more than that, right? So you get a little bit of antagonistic epistasis. Same is true if you do triples, quadruples, right? Most of the things fall along this line or in this direction. You don't see much synergistic epistasis at all. And then you evolve the system So you just do a little gradient descent, very short while, and you can easily find parameters where all the sensitivities are sublinear. This one's sort of close to linear in one direction, right? So this model is much more robust, right? These are not the literature parameters, but these are probably much more biological parameters because of their robustness. And now you do the same thing. You look at pairwise parameter perturbations, and look what you see. A whole little cluster out here that has a much greater effect than would be predicted by just adding up the effects of the individual perturbations. And the same thing for triple, and the same thing for quadruple. And just to connect this to the reality of the output of the model, right? Remember, this is what the model normally does. Here's some of the ways it breaks, right? The model fails to continue oscillating when you turn off the lights, or the model doesn't undergo oscillations at the right period, right? It's got this sort of period doubling thing going on. There are many other ways it breaks, but you get all these failures that are much bigger effects than were predicted. And it turns out that same thing is seen no matter what model you use, right? Except a perfectly linear model. You won't see that. But even if a model is just a bunch of random quadratic equations, you'll see this, right? You always build up these synergistically epistatic cases simply by selecting for robustness in the crudest possible way. Now, population geneticists say, oh, okay, so it can happen, but would you ever find things like that in a real population? So you can do experiments where you sort of seed a population with these alleles and do Monte Carlo simulations and allow them to be selected against. And what you see is initially there's some selection against them, but when their frequency gets down to about 10%, right, selection slows way down because they have to be combined. You have to have combinations in order to have a phenotype you can select against. And so they tend to hang around for a long time. And if you really want to be an evolutionary biologist, some lines didn't come out. It doesn't matter. If you want to argue that, okay, they'll hang around, but will they actually arise in the first place? Will combinatorially fragile, as we call them, alleles arise spontaneously? And so that's easy to show with a very simple model. Sorry for the things that didn't come out in the right color, but this is just A plus B goes to AB. It's just simple binding, mass action binding, right? And so this is the level of AB as a function on a log plot of A and B, right? So this is what the phenotypic landscape looks like for that very, very simple model. And so if we say that having some level of AB is the phenotype of interest, this is what the cells are seeking, right? This would be the optimal phenotype. Normally, the lines of natural selection will push you to anywhere along that intersection of this plane with that surface, right? Because they're all equal phenotype. If we put a little bit of noise in the system, right, we just say that these things, levels can fluctuate, maybe due to environment, right? So if they're fluctuating slow enough, then you can show that the lines of selection draw you right to the corner here. And the corner here is where there's maximum epistasis, right? Where a combined effect of A and B pushes you way up here, whereas an effect of A or B doesn't do nearly as well. This is actually related to the phenomenon of bet hedging. This is the bet hedging position. You don't know which is going to vary, A or B, so you kind of split the difference and put yourself over there. And so you can show with population simulations that they indeed attract themselves to this general area with alleles going out in both directions, and then you pick up these combinatorially fragile pairs as a result of that. So this is just saying this sort of thing arises spontaneously. If it didn't, you'd be really surprised because it's just a very simple geometric effect. Now, one of the nice things about showing this in this way is then you can go back to those models and say, okay, so we got a set of cases that were really fragile that just popped out. Which parameters did they use, right? Which were the genes in which combinations became fragile? And so we can look at that by looking at the landscapes along each of the axes, right? I can't show you a 22-dimensional cube, but I can show you the projections on each of the axes. And the pink zone is where we optimized. In other words, we demanded that the sensitivity be as low as possible in this region. That's how we did the evolution. And so here's all 22 parameters of this particular model. Which ones do you think are the ones that are combinatorially fragile? You put them together and you get big nonlinear synergistic effects. Any guesses? Okay, I'll show you. These are all the combinatorial fragile ones. All right, they all have a very common characteristic. Interestingly, this is the parameter with the greatest influence, right? The most influential parameter has the least to do with the big phenotypes. The parameters that have the most to do have essentially no influence over the regime in which they are normally perturbed. Okay? It's only in combination that they seem to do much at all. It's just an artifact of the fact that the landscape is probably kind of smooth, right? And so therefore you tend to see that. Okay. So I think that's kind of a very strong example of how influence will often lead you astray, right? Focusing on the thing that's most influential would not have revealed any of those combinatorial interactions. But if influence can't tell you what's important, are we stuck? Is there anything else we can do? Is there any other approach? And it turns out, yeah, there are lots of approaches people have considered and are considering right now. And so we'll take a minute to sort of evaluate some of them. So this is one I knew nothing about until I talked about some of this at a meeting. And someone came up and said, oh, I have the answer to your question. It's about the Shapley value. So if you've never heard of Shapley, then you're in the same ballpark. This is the same place I was when I heard about this. But anyway, he's a Nobel Prize-winning economist at UCLA. And he works in the area of game theory, and particularly what's called coalitional games or team games. All right? And he dealt with the question of how do you measure how important a player is in a team sport like, you know, football or cricket or whatever. Right? He wanted to have some measure of the player's importance. And he realized if you just say, what would happen if I took the player away from the team that he happens to be on, that wasn't good enough. Because that's very contextual. That's his importance in that particular context. He said what you have to do is you essentially have to calculate the marginal influence of the player on the outcome of the game averaged over all possible perturbations. That is, all ways in which the player could be on or off every other team. Right? So if you just calculate all those and average them together, then you get the sort of real importance of that player to the game as a whole. Okay? And so that's the Shapley value. And the person who turned me on to the Shapley value is this guy, Eitan Rupin, at Tel Aviv University, who's the one who said, oh, I've got the answer to your question. It's the Shapley value. And he was interested initially in neuronal multi-perturbation experiments. You know, so much of the brain has been mapped by sort of putting a gash in one region or another, and he was curious why no one had ever put a gash in two regions, you know, and whether what you learned from doing it one at a time was really going to be predictive of what's going on in the real system. Ultimately, he moved on to addressing this genetically, and he asked, is the same true with knockouts? Is what we learn from knockouts actually predictive of the real landscape of the genome? Or can single knockouts accurately single out gene function? And so what he did is he used a system that essentially had all been reduced to in silico, right? Basically the entire yeast metabolic system that he had a model of. And he said, okay, I know the phenotypes of knocking out every gene one at a time. But now I'm going to calculate the Shapley value of every gene. That is, what its importance is considering all possible combinatorial perturbations and see if that corresponds with what we learned by the single gene knockouts. And he said, you know, basically he found out that single perturbation analysis misses at least a third of the genes that contribute significantly to the growth of the organism. And so he said, okay, well, this is your, you know, here's your answer, right? If you want to figure out the importance of something, just calculate the Shapley value. And I said, well, that's great, but you had a model already. You knew everything, right? You knew all the, you know, how everything was connected, so you could just numerically calculate the Shapley value. I'm trying to figure out what the model is. I just have a pile of data. He says, no problem. You just sample. Let the computer do the rest, right? You just sample some perturbations, a pair of this, a pair of that, a pair of that, and then you ask the computer to reconstruct what all of the pairwise and triplewise and so on combinatorial perturbations would do. And at which point I realized, well, wait a minute. we're back in the problem we had before. Because if you have control, most of those pairs and so on may do nothing, right? It's going to be the rare perturbation or pair of perturbations that's going to actually reveal the importance of something. So you can't just trust sampling, because you have no idea how deeply you'd have to sample to make it work. So great in principle, if you already know the answer, you can use the Shapley value to convince yourself that you know it. There are a lot of other approaches.
So how do you actually compute the Shapley value? How much sampling do you have to make?
Well, in his case, it wasn't sampling. He was able to compute it.
No, that's right. But
how would you do it if you sampled? You would have to sample and then use some algorithm like Bayesian inference to try to fill in all the things you didn't sample, and then compute from that. So it's very dangerous.
No, what I'm asking is, can you estimate how much sampling would you need to do?
No, you can't, because how deep you'd have to sample is dependent on what the actual landscape looks like. And that's what you don't know a priori.
But say that you want to calculate the Shapley value on your system. So can you estimate whether you will be able to do that or not?
Only if you make assumptions about the system. If you assume, for example, it's a linear system, yes, you can figure out exactly how deeply you'd have to sample. But if the system can have any landscape you like, and particularly a selected landscape that's selected to make it hard to find certain correlations, well, of course you can't predict how deeply you'd have to sample.
So effectively, if I'm not getting you wrong, you're saying that the Shapley value is of no use in this
right. I mean, you can do it, you can do all you want, but there's no way to know that what you've learned is right, right? Because you'd have to assume the thing you're trying to prove in order to know that what you did was valid. Right.
But surely Shapley used it for some...
Oh yeah, but that's not for biology. Now there we're talking about, you know, economics and other situations where you actually have piles of data and you have reasons to think that the underlying model has a certain structure, right? That's the difference. If you know the structure of the model, you can go with estimation. But if you don't know the structure of the model, it's very tricky, you know, to make the assumption that the model is built this way and then assume that the answer you get is going to be a valid one. Okay. Yeah?
So you said in the previous slides ago that you want to look for the influence of that particular parameter gene by looking at what its influence will be in various other networks of a kind, various other systems. So here we are looking at synthetic lethality or synthetic sickness of a kind. So does that answer that question of what the role of that particular parameter would be in various other conditions? Or is this a workaround to get there?
Well, it gets you closer to the answer, right? But I mean, it may be that a pairwise synthetic lethality is just the tip of the iceberg. I was recently talking to a yeast investigator who has a phenotype that's a 5-way epistasis. 5 alleles, different genes, have to be in place to get the phenotype at all. Any subset of that, no detectable phenotype. How you would discover that, you know, if you didn't already know it was there, it's a phenomenal problem. Anyway, let me just talk a little bit about some other obvious approaches that you might think of as ways to figure out what's important, right? This is a method called network inference by multiple regression that originated with J.J. Collins, and it's been elaborated into other things like structural equation modeling and so on. But it's based on a very simple mathematical concept, which is that if you have a system that has a steady state, right, that you can always linearize it for about that steady state, and it will be accurate for small enough perturbations, right? And so therefore, if we probe a system by making lots of small perturbations, one at a time, right, to different things. If they're small enough and we measure the output, of doing that, we should be able to reconstruct the whole system. Right? And especially if you assume a certain sparsity and so on, it should be relatively easy to do that. And they did some experiments in bacteria to suggest they could at least partially reconstruct a network using, essentially a linear approximation and very small perturbations. Turns out there's a problem. And the problem is what I showed you earlier about the kinds of alleles that are fragile in combination, right? Look at their landscapes. They're not just linear about the steady state, they're flat, which means that the perturbations have no effect. Once the perturbations have enough of an effect that you could detect anything over noise, you're outside the linear regime. And so the assumption of the network inference by linear multiple regression completely goes out the window. So it's in precisely the cases you want to find that the method doesn't work. There's other approaches, right? You'll see people using Bayesian network inference. How many people are familiar with Bayesian inference? Few people, right? It's big in computer science. And I'm not great at explaining it, but basically it's a way of finding the maximum likelihood set of connections that would be consistent with a pile of causal data. Data saying that when you do this, this happens, when you do this, that happens, right? And you figure out which connections are most likely to produce that set of data. There's a problem with Bayesian network inference that people rarely make a point of when they use it, which is that it assumes acyclic graphs. That is to say, no feedback loops. Now, how ridiculous is that in biology, right? And the reason you have to assume no feedback loops is because once you have a feedback loop, it's impossible from static data to attribute causality, because you don't know who caused what, the chicken or the egg, right? So now there are modifications to Bayesian network inference that try to infer causality within feedback loops by using time series data. And that's very popular now, time series Bayesian inference. And there's a related method which actually also comes from economics called Granger causality, which is really the same thing. Both of these methods basically use this, they look for changes in one thing, sort of fluctuations that then are correlated with later fluctuations in another thing, and then that gives you the power to figure out which way the causality goes. Okay, Granger causality analysis tends to be used more when you have very dense time series data. So this is actually a little review piece that Chuck Stevens wrote about a paper from Eve Marder about the pyloric ganglion of the crab. Okay, this is a bunch of neurons that make the crab stomach pump so that it can move food down. And the way it makes it pump is these neurons fire in this very interesting rhythmic pattern. And with electrophysiology, you can get very dense data on that. And it had already been worked out from anatomy and other experiments what the network was that was creating this pattern, that there's essentially 3 classes of neurons, and there's a double negative inhibition here, and then this inhibits that. And you can easily see how that would produce an oscillatory circuit. What Eve Marder asked was, if I used just a pile of data of these records and Granger causality analysis, would it find this network, right? Would it figure out what the patterns of influence were? And the answer is no, that it always found a wrong connection instead of this right connection. It said PY inhibited AB instead of AB inhibiting PY. And so they wondered, why did it get it wrong? And so they built a model of this and instantiated the model with a bunch of parameters and then did the Granger causality analysis on the model outputs. And what they found is whenever the model produced chaotic behavior, or maybe not chaotic, but irregular behavior, Granger causality analysis worked like a charm. But with the biological parameters that enabled it to produce rhythmic, beautiful behavior, that's when Granger causality analysis failed. Right? Now, Granger causality analysis is not perfect. No one ever said it was. But the important thing here is it says that it's most likely to fail when the system is operating well. And it's for the same reason. This system is under control. The parameters here allow the system to be under tight control, well coupled. And that deprives Granger causality analysis of correlations between perturbations anymore, and that's why it screws up. Okay, so all of those are just to tell you that the ability to infer importance solely from measurements of influence is specifically defeated by the presence of control, right? It's control per se that makes it hard to do this task. Yes?
So I'm a little confused here because I'm not sure how you're measuring importance. So that seems to...
I'm not.
No, but
Yeah.
how you are defining
it.
importance.
Yeah, that's
So
true. I've skirted that issue a little bit, right?
It
Importance
seems to me
is that which you need to know in order to say you've understood the system. So that's the connection I'm saying, right? If you have a big pile of data, not all of it matters to your understanding of the system. And so effectively I'm saying understanding the system is not the same as being able to predict a pile of phenotypes, right?
Right, but if you're talking about a system where you already know what that system is for, so, but you can run across situations where you don't even, where not only do you not understand the network, you don't even know what the network is supposed to do.
Exactly, exactly. So I guess my point is what we want to learn in biology is what it's for, what does it do, what its function is. And what I'm saying is if you don't know what the function is at the beginning and you approach it in this way and say, well, just let the data tell me, the data won't.
But in that case, what are you even measuring? What's the thing on which you're measuring robustness and...
Oh, with respect to a phenotype.
Exactly. But
Right,
you may not know what the phenotype is.
Well, you always know what the phenotype is, because phenotype is just what you observe. That's the definition of phenotype, variation that you observe. But phenotype isn't the same as performance. Performance is what the organism cares about. Performance is what it's for, what it was built in some sense to do. And really, this is all about, can you infer that from a pile of data? And what I'm arguing is because so much of what things are meant to do is about control, that fact by itself makes it very hard to do the inference. And you can't do it by the ways that are commonly advertised to work. Okay, that's the bottom line. Essentially, can the data speak for themselves? I'm going to argue the data can't speak for themselves. That the notion that big data rescues us from the problem of prior knowledge is completely wrong-headed. Prior knowledge is exactly what we need in order to handle the big data. Right? We must approach it with prior knowledge. And in fact, there are people in systems biology who are sort of seeing this empirically. So there was a nice review article by Trey Eidecker and Lee Hood in which they looked at a number of cases in which inferences were made from big data sets. And they pointed out that critical to being able to get the data under control was to actually have a hypothesis at the beginning and some prior information, expectations about what should be going on with the data. And so that leads me to my final points, which are more practical points. So assuming that one of the things we should take as prior knowledge is that biological systems are very often control systems, and so we should expect control to be there, how should that change the way that we operate as biologists when interacting with big data? And so I'll give you a number of points that will be sort of stuff to take home and think about, because I can't say that I've actually solved this problem, but I have some ideas. So the first one is we really have to pay attention to the real perturbations that influence system performance. We can't ignore that. We can't say, I'm going to study rough colonies, right? It's like the organism couldn't care less about whether colonies are rough. Colonies don't even happen on dishes in the wild, right? We need to have respect for what actually drove evolution in order to have prior knowledge about what should be under control. The reason I put this up, the tightrope walker here, is imagine if you were a Martian, looking down from space at this guy. You'd have no way to know what the real problem here. Well, you could think, oh yeah, human beings walk along ropes across buildings, no big deal, right? Because he does it so well, right? But you and I know, it's breathtaking just to look at the picture because you know how hard it is to balance. So you know what the perturbations are. You know that the human body has trouble with this kind of task. It's actually relatively easy to build a machine that doesn't, right? But you know that the human body has trouble doing this, and so you find this an amazing task. And if you were to ask to find what's important about this task, you'd immediately go to his balance system and how he's balancing. And you'd understand what this rod is for, right? Because of your prior knowledge of what the perturbations are. And so the question is, can we do the same thing when we're faced with something like this, right? What's the hard task that this is doing? We really have to think about that rather than just try to crunch that and find all the patterns of influence. Okay, number 2, I think we have to focus a little more on principles of how control works in biological systems. So I know there are some engineers in the audience. Can anybody tell me what that is? I don't want to put you on the spot too much, but close, yeah. It's a voltage regulator, right? Okay, and the thing is, if you're an electrical engineer and you actually work with these things, you'd know right away it was a voltage regulator. I can't tell you why, because I'm not an electrical engineer. But the real question is, how would an electrical engineer know from the wiring diagram that it's a voltage regulator? Not the way a biologist would find out, which is to do experiments, right? You wouldn't go in and take parts out and learn that it regulates voltage, because in fact you wouldn't learn that. You'd learn that things change, but unless you thought to check that it was a voltage regulator, right, you wouldn't actually find it out. But an engineer can say, oh yeah, because that's how you'd build a voltage regulator, right? They'd see the design principles. They'd say, oh yeah, this has just what you need and not too much more, right, to do a good job of regulating voltage, right? So it's recognizing the design principles in the structure that gives you some prior knowledge to ask the right question. And so I think we have to do that more in biology. And just to give you an example of really simple design principles, right? I mean, suppose the task is you just want to increase A really fast, right? Most people who are not too quantitative minded, so I don't mean the people in this room, but most biologists will say, well, make A at a faster rate. And that's the wrong answer, right? Because the steady state relaxes at the same rate, regardless of how fast you make A, right? You just increase the rate. It doesn't make a difference. What you have to change is the rate at which A turns over, right? If A turns over quickly, it comes to steady state fast. If A turns over slowly, it comes to steady state slow. Now, if you put a feedback loop of A onto V, then it looks like this. Depending upon the gain of the feedback, you can now make A come to steady state very quickly, even if it doesn't turn over very fast, right? Because you can start it off going at a very fast rate and then level off after that, right? So, in effect, there's a design principle. Negative feedback is a generic strategy for making things happen fast without incurring a lot of wasteful fluxes. You're not constantly throwing this stuff away. It's a trivial design principle, but it's there. And every time you see a negative feedback loop, you should ask yourself, is that why that feedback loop's there? Right? And so that's just an example of one way you can approach the structure of these big data models and look for motifs and so on. And of course, there are lots of people in systems biology who've defined all kinds of motifs and operational sorts of things. We even heard about bang-bang control from, who talked about bang-bang? I think Boris may have mentioned. No? Was it Boris? Oh, the throwing talk. That was it. The throwing talk had bang-bang control. Okay. Third thing, we have to anticipate the presence of control even if we lack any evidence for it, right? Because if it's possible to control something, it's very likely the evolution will have found a way to control it. If you remember, I talked about this conversation between Wolpert and Crick, where Crick said, oh yeah, we figured this out at diffusion and so on. I said one of the problems with Crick's answer to this was he didn't consider the possibility that the rate at which morphogen was produced could be adjusted by the system. He just said, simplest model, morphogen goes in at a constant rate. What's the rate at which the gradient forms? But if he had been thinking about control, he would have said, well, if the organism wants to get it there fast, it'll start off from a fast rate. And so all of this goes out the window. And another way of putting that is, in biology, explanations that neglect the presence of control are not the most parsimonious, even if they're more complicated, even if they're simpler, right? So basically, Here's Occam's razor in the biology form. Keep calm and expect control. Okay? A model is not a better model just because it's smaller. If it's smaller and it neglects control, you probably have it wrong. And you have to go look and see if that's the case. Okay. The other thing is when you have control, there are patterns of influence internally within a system that you may be able to look for. So for example, in this simple diagram, if you have perturbations, but you keep your performance flat because you've got control, then somewhere you may have a control signal, and that control signal will be correlated with the perturbations. The output won't be correlated with the perturbations, but the control signal would be. So if you think about that genetically, that means you should go and do an experiment, right, in which you look for something that isn't correlated with performance and attribute a crucial role of that thing to the performance, right? I mean, that's, people would think that's crazy. But if you know something about the perturbations, you find something that's correlated with the perturbations, connected to the performance, but not correlated with the performance, that's a sign that you might actually be on to something really important. And then finally, this anticipate patterns like this in phenotypic landscapes, right? The idea that in genetic landscapes, even in genome-wide association studies, where we don't have the statistical power to find the pairwise and triplewise nonlinear interactions, right? We should expect that nevertheless, underlying those interactions should, at least in some non-trivial number of cases, you should have this sort of phenotypic landscape associated with the individual genes underlying it. And that's interesting because people have tried to rein in this problem of genome-wide association and detecting the pairwise interactions by doing things like looking first for the genes that have marginal effects, that is, effects on their own, and only doing the pairwise testing among those and using that as a trick to get rid of or to reduce the multiple hypothesis testing problem. That's actually a very common trick done in genome-wide association studies, is you only look at the things that have some marginal effect, and then you look for their combinatorial effects. And of course, the sense behind that argument is most of the genome's uninvolved in whatever you're looking at, and this way you'll get rid of all those, right? But that's effectively saying you should focus on this to look for the pairwise interactions, whereas I just told you in the toy systems we worked with, you should be looking for something like this. And so the question is, who's right? And it occurred to us you could look for something like this in genome-wide association studies because we're diploids, and there are both heterozygotes and homozygotes for every allele. Not as many homozygotes if the allele's fairly rare, but there are some in these big populations. And you can ask, for those cases where there's a significant homozygote effect, that is nonlinearity as you go up this direction, right? Everyone knows what homozygote and heterozygote means? Heterozygote means one copy, and homozygote means two copies of the allele, okay? So we all have that. And this is related to the concept called dominance as well, that there's a dominant effect of having two copies. But anyway, you should be able to see hints of this in genome-wide association data. And so we'd argue that when you find that, you're more likely to have picked out an allele that would then be combinatorially fragile with something else. And so we actually have been trying to do that. And we've been working with a very small GWAS data set, which you can get away with because it's a special set where all the allele frequencies are pretty high. It was done in mice, not people, but it involves 44,000 markers and not a lot of linkage disequilibrium, and it's been genotyped for traits related to blood pressure and blood lipids and blood glucose and so on. And so you can ask, about the single associations and the pairwise associations. And basically what we do is we take the data and divide it initially into two independent partitions. And in one partition, we look for gene pairs that show the potential for synergistic epistasis. But remember, there's so much noise in the data, you don't know how much of that's real, right? So you can do what's called a permutation test to estimate how much of that would occur at random. You just shuffle all the data, right? And so subtracting this from this gives you the sort of the true, at least the number of true positives. And then in the other partition, we look for these homozygous effects. And now we're going to ask whether if you condition on this, on these genes over here, do you increase the predictive value that you get over there? And the answer is you do. And here the criteria involve, having having somewhat higher confidence that there actually is combinatorial fragility, but even then the true positive rate is trivially small. But now you're going to add in that the homozygous effect is going to be present along with that, and they're both going to go in the same direction, and you can actually get a small number of cases in which we estimate that at least 40% of them are true positives. So at that point, you can take that very small number of cases, you can do another GWAS or some other kind of study to really see if these things are interacting combinatorially. Interestingly, if you do exactly the same study, but now you're going to use the marginal effect, the effect of each individual SNP, rather than this homozygous effect. Just as we predicted, as soon as you do that, you lose all power to predict the synergistic epistasis. In fact, you end up predicting antagonistic epistasis, but you lose all statistical power when you do that. So in fact, it's not surprising that most people who use that as a criterion aren't finding epistasis in these data. Okay, so I've talked a long time. I will finish up with some summaries. The first is that the big data era is upon us. There's nothing we can do about it. But I think it holds a lot of promise for biology. The second thing is the route from big data to understanding requires that we have a sense of what's important in the big data. We really can't just take a pile of data, stuff it in a computer, and get understanding out the end. Data do not do this themselves. It requires the use of prior knowledge. The prior knowledge can be in the form of specifics, but it can also be in the form of general principles or general expectations about landscapes. And the view that biological systems are very often control systems actually provides a rich source of useful knowledge about dynamics and structure that hopefully will enable us to get these problems under control. And so my feeling is, in systems biology, we really need to develop and tap that knowledge. And so I'll just finish up by saying that the work I talked about related to genome-wide associations has been the labor of love of a graduate student and a postdoc working in my group and a collaborator at the Ronan Institute in New Jersey, John Wilkins. So thanks a lot.
Going back to one of the first examples that you have given, the EGF network that is growing from, say, 15 to hundreds. Okay. So what's the scale of the prior knowledge that we have to put in? So is that like thousands of interacting genes, or is tens, or?
Well, what's the scale of the prior knowledge? I don't know. I mean, the prior knowledge I'm most interested in applying is at a higher level. It's sort of more knowledge about structural expectations. We should be able to look at that and say, over here you have what looks like a certain type of feudal cycle or oscillator, or this, we now know that in a lot of EGF signaling there's fold change detection. So you say, okay, this part, that's fold change detector. So to me, that's the kind of prior knowledge that I would most like to bring to bear on these systems. But you could also bring to bear sort of explicit prior knowledge that you know this is important and that's important. The problem with that is a sort of chicken and the egg problem. How did you get that prior knowledge? Are you confident that those are really the most important things, or are they in fact the seat belts on the A380, which are necessary but irrelevant?
Well, actually, I'm asking this because you have spent quite a long time convincing us that robustness is connected to complexity, effectively, to large networks. So one would expect large networks and therefore a lot of prior knowledge to be needed, not just modules.
Right. Yeah, maybe. Yeah. But I mean, I don't know. I'm hopeful that there are ways not to have to approach the problem as a sort of a pile of low-level prior knowledge, but maybe there are higher-level sort of more emergent features of the prior knowledge that we can use as constraints. I don't know. I mean, to me, that's the key research problem, is to figure out how to formulate the constraints so they will be useful for us in parsing the big data. So I hope you guys will do all that.
So a very insightful talk, I must say. My question is that, does this approach rely on some sort of modularity in the system? Because where there is large-scale pleiotropy, would it be possible to say that certain structures are for certain purposes and for control, let's say?
Yeah, I mean, effectively, yes. Assuming that we can look at sub-features in the system and have those be sort of revealing to us is essentially making the modularity assumption, that the system can be broken into modules without destroying too much of its important structure. And honestly, in most cases, we just make that assumption on faith. Now, there are experiments people have done, in silico experiments, that suggest that modularity will evolve on its own. Okay? But I would take that still in the realm of conjecture at this point rather than a proved fact. So I think mostly we rely on the modularity assumption, because if not, our hands are tied. Right? It's very difficult to know what to do if we treat the whole system like one big neural network where everything is interconnected with everything else, and there's no way to subdivide.
Sorry, can I ask another question?
Sure.
So this comes mostly from the bacterial regulation kind of context. So there it seems that there is some sort of, let's say, controversy between a selectionist sort of point of view and a neutral evolution kind of point of view, that many features have just evolved because of some sort of neutral processes.
Yes.
So how would you, I mean?
Oh, well, I mean, I wouldn't go against the idea that, many features evolved not because of selection. I mean, I think that's a fact, right? So consequently, I'm not arguing that you can explain everything in a biological system with a selectionist explanation. Rather, that simply when you have a pile of big data and your goal is to try to make sense out of it, this is one way to try to organize the data, right? And for those things that aren't associated with selection, that really are just random walks of networks, for those things it may be some of these other methods like linear regression, influence, and so on may actually do very nicely, right? So it's the combination of the two that I think we need to do.
Yeah. So looking at an input-output system, you can probably fit a lot of models.
Yes.
Now, for every model, you would have an expectation of the components that are playing role in that system. Mm-hmm. But that is within a parameter regime or in environmental conditions that they do play a role. But what about the components that would start to play maybe a more important role, quote unquote, when the parameter or the environmental regimes change?
Yeah.
So how would one go about expecting what is the model to look at when you're looking at the data?
You could build a model that has a fluctuating environment built in, and people do that, right? So that's just part of, it's now multi-input, right? You have the input and then you have the environmental fluctuation as another input, but you can still treat it as a model. It just becomes, more complicated.
But the components that you are unaware of that may start to play a role.
Right. Well, so if you expect, right, if you know, say, there's a fluctuating environment and that has to be compensated for, right, then you can try to learn what are the design principles that enable you to compensate for it. You can go look for those things.
Okay. Any other questions? If not, I have a couple of questions.
Okay.
You talked about studying causality, right? Causal networks and so on. Mm-hmm. So how useful have they been in identifying control structures in the system?
In biology?
In biology,
Not
yeah.
very. There are these dream challenges and things where people try to infer stuff, and lots of algorithms out there that work marginally better than each other, but none of them work very well.
Okay, the second point is about the predictability that you said. So predictability, maybe you can make a comment on this, it doesn't have to be a single kind of a thing. Predictability can come from machine learning or GWAS kind of association studies. it could also come post-understanding of a bottom-up approach and so on. So
yeah, I mean, so predictive power is a very good test of your understanding, but it's not the same thing as your understanding. It's what your understanding can generate. Right? And you can have predictive power with no understanding.
With
as
no
well.
understanding, that's right.
So imagine that you want to predict these epistatic effects or higher order. So you should look for having a hypothesis, a null hypothesis that the individual mutations are essentially neutral. Should be one way to fish for them.
Well, but I mean, that's a very difficult thing to fish for because most of the genome is unrelated to a process. So its mutations will also be neutral with respect, you know, to the phenotype you're interested in. So how do you distinguish the 99.9% of the genome that's irrelevant from the little bit that's connected but still has no phenotype? Well, so I kind of hinted at one way, which is if you already have some idea of the network diagram, and you say, well, I should look for something that looks like it's centrally in the network and yet has no phenotype. That should be a hint that you're right on the right track. So remember I said about Sark, how people were jumping out the windows when Sark was knocked out and nothing happened. But essentially, I've just told you that that should have been the sign that Sark was really important, was because of how central it was in pathways in which nothing happened. Right? So anyway, that's a conjecture, right?
One would look at the response to perturbation, say measuring expression levels upon perturbation, and those genes that change expression level, even so that should be the
right.
The
so
control should.
Yeah, if you look internal to the system rather than just at the phenotype, right, then you should have the power to detect all the connections, right? The problem is, if you go at that with no hypothesis, then again you get stuck in the multiple hypothesis testing problem, because now every connection is possible, and you have to infer, you know, every possible connection, even the ones that have a combinatorial effect on each other. So you get into that Shapley value argument, right? That can you really infer this by sampling if the system may actually have been selected so that the cases in which, you know, things have a combinatorial effect are going to be very rare and sparse. So it's a difficult problem. Do you know what I mean? If you can make all the inference you want, but you're still restricted by the model that you used when you made the inference. And most of the time the model is a linear model, by the way.
No, but in practice the importance may be inferred from the response to a perturbation, because
I don't know. You know, again, the seatbelt example of, you know, those seatbelts would be judged hugely important to the flight of the airplane when, you know, anybody knows they're not, right? Because you perturb the seatbelts, you take them away, that airplane will not take off, right?
No, you perturb the environment in that case, not the system.
No, you cut the seatbelts. If you cut the seatbelts in half, the airplane will not take off. Right? This was the analogy that Rob Phillips used the other day. If you cut those seatbelts, the airplane will not take off. Therefore, the seatbelts are hugely influential on this phenotype.
But you would realize that because no matter what's the change in the environment, the seatbelts will not respond to that. So they would be absolutely neutral to the changes in the environment.
The seatbelts.
So you will understand that they are unimportant because
this is the argument that importance is associated with a regulatory effect, that things are changing in a continuous manner with respect to environment. That's a hint that those may be regulators, whereas something that's just necessary, right, that may not be. Yeah, actually there's a nice article by Jamie Davies that tries to make that point about why knockouts are a bad way to look at genetics. I think that's one thing you can go look for. It doesn't have to be that way. Something doesn't have to be a regulator to be important. But yes, that would be one example of taking a bit of prior knowledge that you should expect a regulator to look like this and go look for that.
But the regulator isn't by definition something that controls the response. Otherwise, what is regulating? Is regulating against some change.
Right. Right. No,
It's
absolutely.
regulated the response to a change.
And control isn't the only thing that leads to the prior knowledge. I just wanted to make the point that control is one thing we should expect, and we typically don't. And when we expect it, then that should give us constraints on what we expect out of systems. But it's not the only constraints. Right? There are physical constraints, there are historical constraints, what's possible to evolve to, and so on. In theory, we can use all of these as a way to approach the big data. Okay.
Provided the network structure is known, what is the difference of information we obtain from Shapley value and sensitivity coefficient for multiple perturbations in terms of system response?
I don't know exactly mathematically how they differ. I mean, the Shapley value is kind of like a weighted average over sensitivity coefficients and perturbations. But mathematically how that works out, I'm not sure. You'd have to look up the way Shapley values are calculated.
Okay. So Mukund has an announcement to make.